there, I didn't see it get used. Okay. In some cases, a phosphate is cleaved. Okay, so sometimes yes. So let's go back to our question. It's a lame question because sometimes group translocation is yes, sometimes no. So let's go with that one, and we'll probably get it wrong. We'll find out. Okay, submit. Now we got it right. Okay, so group translocation sometimes does use ATP, sometimes not. You saw it in my example, there was no ATP involved, but there was a phosphate. So, all right. Okay, lame question number 2. I didn't look at the answers. When I was picking them, they only saw the questions, they didn't show me the answers. Okay, endospores survive because of what? Yeah, remember dipicolinic acid stabilizes their DNA so they can survive harsh places. Okay. Dipicolinic acid. That was in the notes, by the way. Ooh, clinical case, wow. College sophomore Nadia is a star point guard for her high school's basketball team. She's excited about the division finals on Friday. She's even heard rumors that a professional scout will be in the stands. Ooh, on Thursday morning she wakes up with a sore throat. Her forehead doesn't feel warm and she's able to eat breakfast. So she makes Thursday class. However, when she wakes up on Friday, her throat is noticeably worse. Throat's bad so far. Willing herself to be healthy, she tries to attend Friday classes. Then she gets a rash developing. Tiny bumps on her chest and abdomen. Now she has a fever. Tired, painful to swallow. Oh, I didn't even read this question before. Desperate, she heads to the student nurse with white spots on the back of her throat and on her tonsils. So all right, big game. Suspect she has strep throat because many viral hepatitis. Okay, after the initial exam, the practitioner takes a swab of Nadia's throat for a rapid strep antigen test for group A beta hemolytic strep pyogenes. Hey, we talked about that today, didn't we? That's why I picked this question. The test responds to the presence of particular strep positive when a patient has strep throat. Such as can confirm and help determine what drug is most effective. So where's the question? The results are ready in a few minutes. Beck's positive, blah, blah, blah. Antimicrobial sensor. She'll miss the big game. She'll help them to play again soon. Now where's the question? I didn't see the question anywhere.
It's smaller.
Right. Oh, here we go. She has infection strep pyogenes, which has a gram-positive cell wall. What distinguishes gram-positive cell walls from gram-negative? After all that, that's the question. Teichoic acid, my God, that was silly. Okay, maybe there's more. I can hope they have more here. Continue. Maybe they have more. Didn't I answer it? Okay, submit. Submitted it. Continue. Let's go on to the next item. What's going on there? Anybody want to help me out here?
Maybe your dad's.
Oh wait, there was a question before that. When the nurse looked in her throat, she saw white spots. They're white blood cells. The white spots were white blood cells attempting to fight off the infection. Which of the following statement explains a key difference between her white blood cells and the strep pyogenes infecting her throat? Nadia's white blood cells contain DNA, while strep do not. Is that true? No, they both have DNA. The strep pyogenes have a cell membrane, while Nadia's cells do not. Is that true? No, they both have cell membrane. And they both have a nucleus, while they do not. Yes, that's true. Strep don't have a nucleus, they're bacteria. Okay. Another stupid question. Okay. So after all that, we had 2 dumb questions. Oh wait, there's part B and C here. Which of the following is not a function of the cell membrane? Well, is selective permeability one of the functions of a cell membrane? Yeah. Active transport, is that one? Yeah. Endocytosis, sometimes it's part of the membrane comes off there. And diffusion is when stuff goes right through the membrane. So all those are pretty okay. But let's do endocytosis, because I think that's usually with a vesicle. Okay, let's see what happens. Yes, endocytosis implies there's a vesicle formation, which bacteria don't do. This is a function of eukaryotic cell membranes and not found in bacterial membranes. Cool. That was a harder one. All right, is that it? Okay. What do we have? 43. Whew, that was tough. The molecule that an enzyme acts upon is known as its substrate. Remember, enzymes act on substrates. Carbon dioxide is a byproduct of which of the following? Where do we get our CO2 from? Krebs cycle. Take a look. Krebs cycle gives you CO2. You guys got to study more for next Monday, right? If nothing else, this will tell you should study more. Which of the following can be the final electron acceptor in anaerobic respiration in bacteria? Nah, not pyruvate. That would be fermentation. Yeah, sulfate and nitrate both can do it. Let's try it. Yeah. Sulfate, nitrate. Not, as long as it's not oxygen. Nitrate and sulfate, I gave you those examples of anaerobic respiration. Take a look. It's in week 1 or 2. Okay, 4 more. Which stages of glycolysis is ATP made? No, this is dumb. I don't like this. Let's keep going. Let's show the answer. Okay, I don't like that. No, it's stupid. Dumb question. Where's most of the ATP generated for eukaryotic cells? Mitochondria in the matrix or on the membrane? The matrix is the middle. The membrane is the membrane on the outside. I know they have electron transport on the mitochondrial membrane. So I'm going to guess that one, but let's hope I'm not wrong. That would be embarrassing if I was wrong. Try again, I was wrong. So it must be in the matrix. Yeah, okay. Good job. So I guess the electrons come through, the protons come through the outer membrane to the matrix, and they're saying that's where it's made. That's a bad question. Any question I get wrong is a bad question. You know what I mean? Okay, what's the byproduct of the catabolism of proteins? If you break down a protein, what do you break off? Think about when you pee.
Ammonia.
Ammonia, right? Yeah, I'm pretty sure that they're going to want that. Okay. I think we got an A. I think we blew less than 5, right? We're going to find out. The Calvin-Benson cycle, that's the photosynthesis. Which one's used here? You guys remember? Ribulose bisphosphate. I guess I didn't cover that. Okay, sorry. Okay.
What was it?
Ribulose bisphosphate. It's the most important enzyme on planet Earth. I should have covered it. It's the one that puts sugars together during photosynthesis. It's the most abundant protein on planet Earth. Oops. So remember, that's in your book. So on the test, I might put a few book questions. See who's reading. Okay, because that is kind of important, being that it's without that enzyme we could have no life on Earth. So that might be an important one. What do we want? Last one. Okay, horizontal gene transfer overview. What is this? Click here to complete the activity. Oh my goodness. Like it? Cool.
The transfer of genetic information between organisms in the same generation is called horizontal gene transfer. 3 types of horizontal gene transfer occur in bacteria: transformation, transduction, and conjugation. All 3 mechanisms involve a donor cell that transfers a portion of its DNA to a recipient cell, which may incorporate the new DNA into its own, becoming a recombinant organism.
Okay, I knew that. So, all right, get rid of that. Now, then answer the questions. Oh, they're over here on the right. What's a recombinant cell? It's the one that receives DNA from an outside source and incorporates it into its own. Yes. What is the hallmark of all horizontal gene transfers? What?
Transfer it.
Yeah, same kind of thing.
I don't, all those answers are bad, but we're gonna choose that one,
Yeah.
but they're all bad answers. Another bad question. That one I got right, and I still think it's a bad question. Because they can be, horizontal gene transfer can be between plants and bacteria. They're not the same generation. It's stupid. Okay. What's an example of horizontal gene transfer in bacteria?
Conjugation.
All three of these guys, come on. So there's three kinds of horizontal gene transfer. Transformation, transduction, conjugation. Okay. Continue. All right, so let's see. How do we do? We got